This is the third and final set of notes on Unit 8 about meiosis and karyotypes. Today's topic is crossing over and karyotypes. So crossing over is when homologous chromosomes exchange portions of their chromatids. This can happen in mitosis as well as meiosis. And in meiosis, it results in unexpected gene combinations. So when the tetrad forms, when the two chromosomes, homologous chromosomes, pair up, portions of the chromatids can cross over and exchange portions between them. So you'll end up with several different kinds of gametes. This is important because this is what leads to a lot of the genetic variability that we have within species, because you end up with different combinations of characteristics than you normally would have if there were no crossing over. So crossing over usually occurs in meiosis in prophase 1, although it can occur in prophase 2 as well. Normally we think about crossing over as then the two homologous chromosomes when they form the tetrad, then you have chromatids that cross over, and so you get a mixing of the father's genes and the mother's genes on the same chromosome. So you end up with, in this example, with 4 different, genetically different gametes that can be produced as a result. And again, this is really important to maintaining genetic diversity within a species. As far as chromosomes go, humans have 23 pairs of chromosomes, as you know. 22 pairs of those are what we call somatic chromosomes. These are the chromosomes that are just the normal body chromosomes. You have to have at least 2 copies of each of those 22 chromosomes. So that means you must have those in order to survive. Individuals can be born sometimes with 3 copies of one, but never with 1 copy. The other pair of chromosomes are the sex chromosomes, and they're not necessarily, they're a pair in females, but not really in males. In females, there are 2 X chromosomes. In males, there's an X and a Y chromosome. We call them a pair, but in the male, they're not really a pair because the Y chromosome is shorter and carries fewer genes than the X chromosome. Any sex-linked characteristics or sex-linked genes are usually located on the X chromosome, rarely on the Y chromosome. There are a few sex-linked traits that are carried on the Y, but mostly in humans, they're mostly carried on the X chromosome. So you can take a picture of the chromosomes and line up the individual chromosomes by pairs, and you can see the homologous chromosomes of an individual in something called a karyotype. Okay, they're lined up in order from largest to smallest, and also by the location of the banding shapes that show up on the dying of the cells, and also by the location of the centromeres. This individual, as you can see, has 2 copies of every chromosome as well as the X chromosome, so that makes this individual a female. If it were a male, instead of 2 X chromosomes, there would be only 1 here, as well as the Y chromosome that would show up in this location. Now, sometimes there are accidents that happen when meiosis is occurring, and this is called non-disjunction. Your book defines non-disjunction as an accident of meiosis or mitosis in which a pair of homologous chromosomes or a pair of sister chromatids fails to separate at anaphase. When it happens in mitosis, it usually only affects a couple of body cells, and it doesn't usually cause too much of a problem for the individual. Maybe a tumor that would need to be removed, but nothing major that would happen. But when it happens in meiosis, then it can result in gametes that have either extra chromosomes or missing chromosomes. And this definitely causes a problem for the offspring that can result from fertilization. So in meiosis 1, non-destruction again would occur in anaphase here, where instead of the tetrad separating into 2 chromosomes, you end up with 1 tetrad that doesn't get separated. In this case, you would have 2 gametes. All of your gametes would be abnormal. 2 of them would have 3 chromosomes here, and 2 of them would have only 1. Chances of individuals resulting from fertilization of these -1 would be depending on what chromosome it was that was affected. In meiosis 2, non-disjunction that would happen in prophase 2 would be when you had one of the pair of sister chromatids that did not separate, and so you end up with 4 different results here in your gametes: 1 that has 3, 1 that has 1, and then 2 that that are normal gametes that have 2 chromosomes each. When you end up with 3 copies of a chromosome, that is called trisomy. You have 3 chromosomes instead of the normal pair of homologous chromosomes. An example of trisomy is Down syndrome. This is trisomy 21 because there are 3 copies of chromosome number 21. As you know from your personal knowledge of people with Down syndrome, this causes various kinds of physical as well as mental characteristics that apply. There are several different kinds of trisomy that can that cause abnormal conditions for the offspring. There can also be trisomy of the X chromosome, in which case there really isn't anything negative that happens. The females with 3 X chromosomes do not have any abnormalities that are apparent unless you look at their karyotype. In terms of missing chromosomes, there are a couple of conditions where you can have a missing chromosome, but that almost always, as far as I know, involves the sex chromosomes. So an individual with Turner syndrome has only 1 X chromosome. All of the somatic chromosomes are paired up, but in terms of sex chromosomes, there's only 1 X and no Y chromosomes. This would be a female that has only 45 chromosomes, and there are physical characteristics, including infertility, that result from this condition. And a male can also have an extra chromosome, okay, an extra X chromosome here. In this case, we call something called Klinefelter syndrome, and you'd end up that there are also physical characteristics that show up, as well as infertility that occurs in these individuals. They are male in appearance, but they have some female characteristics as well. It is possible for a male to have an X chromosome and 2 or more Y chromosomes, in which case, again, they're pretty normal in appearance, and everything seems to be normal, and you would only notice when you did the karyotype. This concludes the notes on crossing over and karyotypes. We'll talk more about some of these non-disjunction disorders in class.